Hello everyone, welcome to my channel Bani's Biology. Today we will explore one of the most remarkable DNA repair system in bacteria, that is the RecBCD pathway. This pathway is the bacterial solution to repairing dangerous double-stranded break, and this double-stranded break can be caused by UV radiation, oxidative stress or even program events during genetic exchange. If left unrepaired, the cell dies. So the cell need a way to recognize broken DNA and process them and restore genetic integrity. So bacteria evolve a remarkable machine, that is RecBCD model. So what is RecBCD? RecBCD is a multifunctional enzyme complex in E. coli, and RecBCD also called exonuclease 5, and that repair the double-stranded break in DNA by unwinding and degrading DNA and then prepare it for homologous recombination. So RecBCD enzyme consists of three subunits. One is RecB, another is RecC and RecD. RecD. So RecB have 3' to 5' helicase activity and nuclease activity. So helicase activity means that unwind the double helix DNA. So this is the double helix DNA that is unwound by RecB, and nuclease activity that cut the nucleotide that cut by RecB, and RecC that recognize the chi site, that is a special DNA sequence that regulate recombination, and RecD that have 5' to 3' helicase activity. So together these three protein form the RecBCD. So here is the RecB sequence, RecC sequence and RecD sequence. So RecB that have both helicase and nuclease activity, and a RecD that only have a helicase activity. So here RecB and D both have helicase activity. That means they both unwind DNA from a broken end, and RecB have nuclease activity, that have only nuclease activity. That means RecB cut DNA strand and producing single stranded fragment. And next RecC detect the chi site, that is 5' GCTGG TGG 3' site, and at the chi site point RecBCD changes its cutting behavior to favor recombination instead of degradation. So this is the chi site. Here recombination frequency is 10x. In the B side or A side, here is the recombination frequency 5x and 2.5x, that is continuously decreases, and so recombination frequency is high in the chi site. So chi site help in the recombination instead of degradation. So let's talk about the stepwise process of RecBCD function. So first RecBCD enzyme bind tightly to the free end of a broken double-stranded DNA molecule. So first RecBCD enzyme, this is the RecD, this is the RecC and this is the RecB. So RecBCD enzyme tightly bind the free end of broken double-stranded DNA molecule. This is the broken strand. This is the broken strand. And RecB have 3' to 5' helicase activity. So RecB bind in the side of 3' end. This is the 3' side. And next RecD have 5' to 3' helicase activity. So RecD that bind in the side of 5' side. And these both unwind the DNA duplex. But during the initial phase, the two motor of complex RecB and RecD are not moving at equal rate. RecD which move rapidly on the 5' ending strand, and RecB which moves slowly on the 3' ending strand. Because of these two subunits travel at different speed, RecB accumulate a single stranded DNA loop in the lower strand during unwinding because RecB move slowly. This is the loop formation. So as the DNA is unwind, RecB have nuclease activity that continuously degrade the both strand. Both strand is continuously degraded, but this degradation is asymmetrically. Initially nuclease activity is unregulated and digesting DNA indiscriminately until a chi site is encountered. Then RecC recognize a chi site sequence. This is the chi site sequence. RecC is recognized. After encountering the chi site, RecBCD changes its behavior. So this time RecB stop degrading the 3' ending strand. This is the 3' ending strand. So RecB stop degrading the 3' ending strand but continuously degrade the 5' ending strand. This is the 5' site. So this provide a long, this is the long 3' single stranded DNA tail. So one of the most thing is RecB and RecD. So RecB and RecD. D are ATP dependent molecular motor and require ATP and Mg2+. So ATP hydrolysis provide the energy to unwind the double stranded DNA from the broken end, while Mg2+ is essential as a cofactor that stabilize ATP and enable nuclease activity. So lastly, 3' long stranded DNA tail produces. This is a 3' long DNA single stranded DNA. So then RecBCD help RecA. So RecA is loaded in the 3' single stranded DNA tail by the RecBCD protein. So RecA coated the single stranded DNA forming a nucleoprotein filament. So this is the RecA coated 3' single stranded tail and forming a nucleoprotein filament, So filament grows by the addition of RecA subunit by the 5' to 3' direction. So RecA is coated in the 5' to 3' direction. So such that a DNA strand that terminates in 3' end is most likely to coated by RecA, and RecA filament assembly on the DNA 3' end. This is the 3' end site, significantly faster than do on the 5' end. So this is the double stranded break, and after double stranded break generated 3' single stranded tail. This is the 3' single stranded tail, and 3' single strand is coated by RecA protein. So subsequently the single stranded bounded by RecA perform a stand invasion. This is the stand invasion, and this stand invades the unbroken homologous DNA duplex. So this is the unbroken homologous DNA duplex, and this form the D-loop structure. This is the D-loop structure. Where the invading strand pair with the complementary strand in the other homologous DNA molecule. So this is the strand invades and that pair with the complementary DNA strand of another homologous DNA molecule, and the invading strand end with 3' termini. This is the invaded strand with 3' terminal, and this is also second invaded strand, and this also 3' terminal serve as primer for new DNA synthesis. So elongation is happened from the DNA end. So continuously elongation happens, and using the complementary strand in the homologous duplex as a template, and serve to regenerate the region of DNA that the destroyed during the processing of the strand at the break site. So continuously new DNA is synthesized using the complementary DNA strand of the homologous DNA duplex, and that act as a template strand. So that's how double stranded break is repaired. So after that holiday junction is formed. This is the holiday junction, and which are later resolved by specialized enzyme. So the broken DNA is repaired by RecBCD enzyme complex. So let's summarize the whole process. First, DNA end binding. RecBCD first ideally bind to the end of broken double stranded DNA molecule. After that, RecB and RecD that unwind the DNA. Because of the RecB and RecD have helicase activity. RecD have 5' to 3' helicase activity, and RecB have 3' to 5' helicase activity, and RecD that moves faster than the RecB, and RecB move along the strand with 5' end, and RecB move along the strand with 3' end. And because of the RecD move faster, a loop single stranded DNA structure form. And after that RecB that have nuclease activity that degrade both strand and producing 5' phosphate end. Next, when RecC encounter a chi site, RecBCD changes behavior. Before encountering a chi site, RecB cut the both ends 5' and 3' both ends, but after encountering a chi site, RecB only degrades the 5' end, and degradation of 3' end strand is reduced, and the 3' tail becomes stabilized and extended, and this 3' tail end is coated by RecA protein and enable it to invade homologous DNA, and this RecA coated strand pair with homologous region and initiating strand exchange and double stranded break repair, and that's how RecBCD help in double stranded break repair in E. coli. So in conclusion, RecBCD is a helicase plus nuclease powerhouse that not only repair DNA but also drives recombination. And understanding this model help us appreciate how cell maintain genetic integrity under stress. So if you enjoyed learning about the RecBCD model, don't forget to like, share and subscribe for more molecular biology deep dives.